Okay, so today we are going to be talking about a pure culture. A pure culture is just an individual organism in a culture media. It's not mixed with anything, it's not contaminated with anything, it's like it says, a pure culture. So there are three different techniques that you can use to isolate the organism from its colonies. Those techniques are going to be the pour plate, the streak plate, and the spread plate. Today we're going to focus on the streak plate. So there are a bunch of steps to be able to complete the streak plate technique. Those steps are you're going to have a metal loop and you're going to sterilize it in your Bunsen burner. Then you're going to dip it in your sample that you're testing. For our experiment, we are going to use a urine sample. You're going to want to take your metal loop, stick it in the urine sample, and then you're going to have an agar plate and you're going to want to streak that sample across 1/4 of the agar plate. You're going to take your loop, sterilize it in the Bunsen burner, let it cool down, and then you're going to want to start in the second quarter, streak it into the first, back into the second, back into the first, back into the second quarter. You're then going to streak the rest of the second quarter, and then you're going to take your loop, sterilize it, let it cool, and then you're going to start in the third quarter, streak it into the second, back into the third, back into the second, back into the third, and streak the rest of the third quarter. After those first two streaks for both the first and second quarter, and then the second and third quarter, you do not want the quarters to touch. Then you're going to sterilize your loop again, and you are going to start in the fourth quarter, streak it into the third, back into the fourth, back into the third, back into the fourth, and then streak the rest of the fourth quarter. You do not want the fourth quarter and the third quarter to touch after the first two streaks. You are also going to not want the first quarter and the fourth quarter to touch because the first quarter is a lot less diluted. There are just a lot more colonies, and what you're trying to do is isolate the colonies. So if they touch, your whole experiment is contaminated and you're just going to have to start over. So with our example, we're talking about a urine sample. So we're talking about having a patient, and this is in the clinical setting. So having a pure culture and isolating the organisms are important for a clinical setting because we have to be able to study these organisms to treat the patient's disease. So you're going to want to isolate the organism and study that. You're going to want to study the genome sequence. You're going to want to study the antibiotic susceptibility and the organism's virulence. Studying all of these traits of the organism are going to allow you to be able to manipulate the different characteristics and traits, and you're going to be able to create a treatment for the disease that the patient is suffering from.